Taking a look at enzymes, part 1. Enzymes are globular proteins and biological catalysts, meaning they're necessary for every metabolic reaction in living things. Enzymes fold into the precise shape that is needed. Many of the enzymes' names end in ASE, that ACE ending, such as amylase. Enzymes are going to speed up chemical reactions of life inside and outside of cells without being changed, so only a small amount is needed. Enzymes are used for forming bonds between molecules, such as a condensation reaction, which is also known as dehydration. An example would be the synthesis, putting something together, such as allowing two glucose molecules to bond together. Enzymes are also used for breaking bonds between molecules. This is known as a hydrolysis, and an example would be digestion. Without enzymes, could you imagine how long it would take for your last meal to actually digest? All right, have a question for you. Please take a moment to pause and answer this question. Intracellular and extracellular enzymes. Intracellular enzymes are the enzymes that work inside the cell. Extracellular enzymes are enzymes that are secreted by the cells and catalyze reactions outside of the cell. An example would be digestive enzymes in the gut, or fungi that secrete enzymes outside their body to digest food on which they are growing. Most enzymes are intracellular. An example of an intracellular enzyme would be pepsin or maltase. This chart is just giving you a quick comparison between intracellular enzymes and extracellular enzymes. Again, there are more intracellular enzymes than extracellular. Intracellular occurring within the cell for things such as photosynthesis, DNA replication, cellular respiration, protein synthesis, and intracellular digestion, whereas extracellular enzymes would be needed for extracellular digestion and decomposition. Again, this is just a quick little snapshot. There are others. Also includes some examples for you here at the bottom. Enzymes have hydrophilic groups on their surfaces which bind to substrates. So if these two molecules were going to combine, again, the water molecule is going to be released. This is known as a condensation reaction. In hydrolysis, water is going to be taken in to break the molecules. In order for this process to occur, condensation or hydrolysis, we are going to need an enzyme to help with this process in both cases. Again, looking at an example of condensation where you're going to have a glucose molecule combining with fructose in order to make sucrose. In order for these two to combine, to actually bond together, condensation reaction occurs where water is going to be released. In hydrolysis, remember, you start with the molecules bonded, so we start with sucrose. We're going to need to take in water in order to break this bond to separate out the glucose and fructose. In either event, we are going to need an enzyme to help with the process. Some chemical reactions release energy. These are known as an exergonic reaction, such as digesting polymers. These would be hydrolysis reactions or a catabolism, which is the breakdown of complex molecules in living organisms to form simpler ones. Digesting molecules is less organization. There's a lower energy state. Some chemical reactions require energy, the input of energy. These would be your endergonic reactions, such as building polymers. This would be your dehydration synthesis, also known as your condensation reaction, or an anabolism, which is the synthesis of complex molecules in living organisms from simpler ones, combining them together with the storage of energy. Building molecules, more organization is required, higher energy state. Looking at graphs for endergonic and exergonic reactions, this first reaction is showing you an exergonic reaction where you have your reactants. They have to take in enough energy. Once they have enough energy, then the reaction proceeds to start forming the product. And so this is the energy that's actually going to be released. Again, this is representing an exergonic reaction. Energies released, such as in digestion. And then your change in your energy amount is what's known as Gibbs free energy. You're looking at Gibbs free energy, you're looking at the ability to do work. A negative change in release of free energy would be an exergonic reaction. Notice your reactants are going to be here. That change in energy, if you take that amount straight across, and then your product amount for energy. So that change here would actually be a negative amount. When we look at an endergonic reaction, you notice your reactants. Again, they gotta take in enough energy before the products are formed, but your products are up here. So energy has to be supplied for this reaction. This again is known as an endergonic reaction. Energy is invested into this system, such as synthesis. So here you would have a positive change in your Gibbs free energy. Organisms require energy to live. But where does this energy come from? Combining the exergonic reactions, again, which release energy, with an endergonic reaction, which is needing energy. In this top example here, we're looking at, let's say, digestion occurring. When digestion occurs, again, you're going to be breaking apart these molecules. You're going to get energy that's going to be given off. Think about when you eat food, you digest it, gives you energy. This energy in turn can go into a synthesis reaction where we need to take energy in for the system to proceed in order to combine the molecules together. So what drives reactions? If reactions are downhill, why don't they just happen spontaneously? Why is it that stable polymers spontaneously digest into their monomers? Well, it can't happen spontaneous because the covalent bonds are stable bonds. There's a lot of stable bonds in this structure. Activation energy. Breaking down large molecules requires an initial input of energy. So here we have that cellulose material. This is representing like wood for cellulose. And it's going to burn with energy, which is going to produce carbon dioxide and water and heat. It requires an activation energy. Large molecules are going to be very stable. They require high amounts of energy to break the bonds. Catalysts reduce the activation energy. So when we are looking again, our activation energy is the amount of energy needed to break those bonds of a molecule. So we start with our reactants. Our reactants have to take in energy to the top of the hill, if you will. The top of the hill is where the activation energy is. They have to move the reaction over the energy hill before they can start to form the products. To do this, we need some type of catalyst. We could use heat. That'll help speed the reaction up. But when we're looking at glucose, adding heat to the system would be a concern. If you think of the second law of thermodynamics, the universe tends to be disordered. So why don't proteins and carbohydrates and other biomolecules just break down? At temperatures that are typical of the cell, the molecules don't make it over this hump of activation energy. This being the hump of activation energy. But a cell must be metabolically active. Heat would speed up a reaction, but it would also denature the protein and kill the cell. So we have enzymes that would help with this process to be able to have those reactions occurring faster. Taking a look at biological catalysts, soluble proteins with hydrophilic groups on their surfaces. These facilitate chemical reactions. They can increase the rate of the reaction without being consumed. These are your enzymes. They're going to reduce that activation energy. They don't change the free energy, that change in Gibbs free energy, whether it's released or required. That doesn't change. It just allows things to happen faster. Enzymes are required for most biological reactions, both intracellular and extracellular. Enzymes are highly specific. There's thousands of different enzymes in the cells which control reactions of life. Taking a look at some vocabulary words. We have our enzyme. This little purple structure represents an enzyme, and you'll notice the enzyme shape is very specific to match with the substrate. This is the substrate that's going to actually bond with the enzyme. Your substrate is the reactant, which again is going to bind to the enzyme. And then when they're together, here's your substrate combined with the enzyme. We call this an enzyme substrate complex. It's a temporary setup, okay? It's not going to stay that way. Again, when they're combined together, that enzyme along with the substrate, that's what's known as an enzyme substrate complex. And then you have the product, which is going to be the end of the reaction. You started with your substrate, a chemical process occurs, and then the product is released. And notice that our product is different than our original substrate, which was the reactant. The enzyme is going to go back. It stays in its original shape. It is not going to be part of the reaction. This spot here, you can see it a little bit better on the enzyme, where the substrate is going to combine with the enzyme. This is what's known as the active site. The active site is the enzyme's catalytic site. the substrate fits right inside that active site. Taking a look at some of the properties of enzymes. Enzymes are reaction specific, meaning each enzyme works with a specific substrate. There's a chemical fit between the active site and the substrate, where you'll have hydrogen and ionic bonds. Enzymes are not consumed in the reaction. Single enzyme molecule can catalyze thousands or more reactions per second. Enzymes, again, are unaffected by the reaction. Enzymes can be affected by cellular conditions. Any condition that can denature a protein structure could affect the enzyme. Remember, enzymes are proteins. A change in temperature or pH or salinity can cause an enzyme to be denatured. Enzymes are named for the reaction they catalyze. The names of enzymes usually end in that ASE ending, such as sucrase, which breaks down sucrose, protease, which breaks down proteins, lipases break down lipids, DNA polymerase builds DNA, adds nucleotides to the DNA strand, pepsin breaks down proteins. Globular proteins, enzyme molecules, are coiled into a precise three-dimensional shape. There's the hydrophilic R group, which is a side chain, on the outside of the molecule that makes them soluble in water found within the cytoplasm. Enzyme molecules, again, have an active site. The active site of the enzyme is a region where another molecule can bind. That's where the substrate is going to bind to it. The shape of the active site fit perfectly together, which is known as a lock and key hypothesis. When they bind together, it's known as an enzyme substrate complex. So here's your enzyme. This is the active site where the substrate would actually bond. This is our enzyme substrate complex where you have your enzyme and there's the bind that has occurred between the enzyme and the substrate. And then, of course, once the reaction occurs, then it's going to release the products. The enzyme is not consumed within the reaction. Taking a look at the lock and key model or lock and key hypothesis, enzyme and substrate are complementary. And complementary is a key word that you want to use when you're describing how a substrate's going to fit perfectly into an enzyme's active site. When we take a look, this is our enzyme, this blue structure. The active site is this opening. It's complementary to the substrate so that they can bind together to give us our enzyme substrate complex. When they actually are binding together, that's going to have a hydrogen bond between the two. They'll end up fitting together like a lock and key. In biology, the size isn't important, it's the shape that matters. Remember that each enzyme will only act on one type of substrate molecule. They are specific shapes for the enzyme and a specific shape for the substrate. There's also the induced fit model. In 1959, it was found that enzymes are more flexible than they were originally suggested. Originally, they were believed to be very rigid and locked together like a lock and key hypothesis, but the modern hypothesis actually has more of an induced fit model. Here, the shape can slightly change to ensure a perfect fit. You still have your enzyme. We still have an active site. And then your substrate is still going to combine into the active site, but it's going to slightly alter its shape to allow for a perfect fit. And that's what's known as the induced fit model. When that happens, it kind of starts to break those bonds a little bit so that it helps lower the amount of energy that's also needed. An enzyme may catalyze a reaction where a substrate is broken into two or more molecules. An enzyme could also catalyze the joining of two molecules together, which would be like an example of a dipeptide. So looking back at our picture here, we have our enzyme, and then we're looking at our substrate. It can bind together at the active site, and then when it's combined together, we have our enzyme substrate complex. But when the product's released, notice we end up getting two products as a result here. But the reverse could also be true, where you could start your substrate, there could be two substrates that are actually combining at the active site and then producing one product in the end. This is just a simple diagram showing when you have your enzyme in purple and then your substrates, you have two of them and they're going to bind to the enzyme, again giving you that enzyme substrate complex. And then after the reaction, the product is going to only have one product where you started with two substrates. Again, the enzyme would not be consumed within the reaction. How does the induced fit work? There's a variety of mechanisms for enzymes to lower the activation energy and speed up a reaction. In a synthesis reaction, the active site orients the substrates into the correct position for the reaction. The enzyme brings the substrate closer together. In digestion, the active site binds substrates and puts stress on the bonds that must be broken, making it easier to separate the molecules. A molecule of the enzyme catalase can bind with hydrogen peroxide molecules, split them into water and oxygen, and release the products at a rate of 10 million molecules per second. Let's take a look at lysozyme. Lysozyme is found in tears, saliva, and other secretions. It's a natural defense against bacteria. It does this by breaking the polysaccharide chains that form cell walls in bacteria. The lysozyme breaking a chain in a polysaccharide. In this first diagram in part A, the diagram is showing the formation of an enzyme substrate and enzyme product complexes and releases the products. In B, it's just showing you more with the globular enzymes, the globular proteins, the space-filling model showing the substrate and the active site of the enzyme. The substrate is a polysaccharide chain which slides neatly into the groove of that active site and is split by the enzyme. Many such chains give the bacteria cell wall the rigidity. When the chains break, the wall loses its rigidity and the bacterial cell explodes as a result of osmosis. In looking at activation energy, remember the reactions need to take in enough energy for the reaction to occur. That's what's climbing up to that hilltop. In many chemical reactions, energy is needed for the substrate to convert to the product. For example, you had to heat your Benedict solution before you were able to see the reaction of the sugars. Enzymes lower the activation energy to avoid adding energy. They do this by binding with the substrate to allow for the molecules to react more easily. With enzymes, the reactions are catalyzed by enzymes taking place more rapidly at a lower temperature. So when you're looking at these two graphs, the top graph is showing you a very high hill, meaning a lot is needed for the activation energy. But when an enzyme is present, you lower that hill. So it lowers the activation energy, allowing the reaction to occur faster. Notice your starting point for your reactants and your ending points of the products are still the same. It's just we've lowered the activation energy. Enzymes reduce the activation energy. Catalysts and biological enzymes are able to reduce the amount of energy required to start a reaction. So we have our reactants and we have our products. Remember, they have to take in enough energy for the reaction to begin. So this would represent, this white hill is representing the uncatalyzed reaction. So I have a very large amount for my activation energy. My activation energy goes from the reactants to the top of the hill. But in a catalyzed reaction, adding in that catalyst, it lowers the hill, which lowers my activation energy. This is going to represent my new activation energy, a much smaller amount. Again, my starting amount and my ending amount are still the same. My change in Gibbs free energy is the least amount of energy required or given off, and it can't be decreased by enzymes. So my Gibbs energy is going to still be the difference between my reactants and my products. So this is what's representing my change in the energy here. Without enzymes, most reactions would occur very slowly, which would make it almost impossible for life to exist. The turnover rate is the number of products produced by an enzyme per second. So the enzymes really help because they allow us to lower that activation energy, which allows the reactions to speed up. Investigating the progress of an enzyme catalyzed reaction, measuring the rate of formation of a product. And this is an example that you guys need to be familiar with. Hydrogen peroxide is sometimes produced in cells. It is toxic, so it needs to quickly be removed. An enzyme in tissues catalyzes and breaks down the hydrogen peroxide into water and oxygen. The oxygen is released, collected, and measured. And that's what our graph is showing us. The volume of oxygen that's released in centimeters cubed, the time that that's occurring measured in seconds. The reaction begins quickly. During the first minute, a large volume of oxygen is collected. As the reaction continues, the amount of oxygen released slows down. How come? At the beginning, there's a large number of substrate molecules. The rate depends on how many enzyme molecules there are and the speed at which the enzyme can convert the substrate into product, release it, then bind with another substrate molecule. As more substrate is converted into product, there are fewer substrate molecules to bind with. Enzymes which allow for the reaction to slow down and eventually stop. The curve is the steepest at the start of the reaction, and the rate is the fastest. You can measure the initial rate of the reaction by calculating the slope of a tangent to the curve as it's close to the time zero as possible. I actually have a handout to give to you to show you how to do this. All right, question for you guys. Please pause the video and take a moment to answer this question. Using a colorimeter to measure the progress of an enzyme-controlled reaction. Colorimeters are used to measure the color change by measuring the absorption or different wavelengths of light. This provides a quantitative data that could be plotted in a graph. When testing amylase, which is a type of starch, you can measure the intensity of the blue-black color obtained in the iodine test. This would allow you to plot a curve of the amount of starch remaining against time. You could then calculate the initial reaction rate with the same way that you did for the catalyst hydrogen peroxide reaction. All right, 2 more questions for you guys, if you would take a moment to answer those.